Welcome to the audio presentation of info clustering. The presentation will be about 30 minutes. The word clustering refers to the process of grouping similar things together while separating dissimilar things apart. It is a simple engineering idea that turns out to have significant applications in many areas of scientific research, such as bioinformatics, neuroscience, and network science. These research often involve a complicated biological and social systems that consist of a massive amount of entities interacting with each other. The ability to group similar entities together based on their interactions reduces the system into smaller, and therefore more manageable, subsystems for further studies. Nowadays, there are many technologies available to collect data about the systems. However, given the huge volume and variety of data available, the main challenge is to automate the clustering process using a mathematical criterion that leads to meaningful, yet arithmetically simple to compute, clusters. I believe the key lies in a better understanding of what information is, and how we can measure it quantitatively. To address this challenge, I would like to present a new information theoretic approach to clustering, called info clustering. Let me first motivate the idea of info clustering with some of its fascinating applications. In bioinformatics, an important area of study is the human genome. We know that the biological information of a living organism is encoded entirely in its DNA sequence. In human being, there are 23 pairs of chromosomes, each of which is a long strand of DNA double helix. The double helix, in turn, consists of segments of DNA called the genes. Some of these genes express itself in the form of gene products such as enzymes, hormones, and receptors, and these proteins carry out important functions that sustain the different metabolic pathways important to our lives. In the human genome, there are 20 to 25,000 such protein-coding genes. Two important open questions are, how these genes work together to sustain the metabolic pathways, and how mutations of the genes causes certain disease such as cancer. Clustering is a helpful first step in studying the metabolic pathways and disease pathology, because it helps identify smaller subsets of related genes that work closely together. More precisely, although different genes express differently in different people, genes that are closely related tend to have similar expression patterns. Such co-expression of the genes means there are information mutual to the genes. In other words, if we have a way to measure the mutual information among the genes, then we can cluster the genes according to their mutual information. Another fascinating application of info clustering is in the study of the human brain in neuroscience. We know that the brain carries out important tasks such as perception, emotion, thought and memory. The way it works is that, the cerebral cortex in human brain consists of about 16 billion nerve cells called neurons. These neurons are physically wired together by fiber-like projections. The neurons stimulate each other in some pathway circuitries to carry out the brain functions. More precisely, when a neuron gets excited, it sends an electrical signal down the axon, which branches out to one or more target neurons. The electrical signal may further be converted into a chemical signal that excites the target neurons through the synapses. The stimulation mechanism of the neurons have even inspired a family of learning models in artificial intelligence called artificial neural networks. The artificial neural network consists of layers of computational units that mimics how the neurons stimulate each other. By the deep learning methods in machine learning, such artificial neural networks can be trained to stimulate in a way to perform complicated tasks such as image recognition. The performance can be much better than other approaches. However, exactly why it works so well is unclear. That is, it is not entirely known why neurons can carry out complicated tasks by simply stimulating each other in some pathway circuitries. There are also many mental disorders we cannot explain. For instance, schizophrenia is a brain disorder where the patient fails to understand reality. The cause of such disorder remains unknown, even though there have been observed differences in the brain activity of frontal lobe of the brain. Nevertheless, it is recently discovered that the brain segregates into tightly connected regions, and there are important network hubs, called the rich club, that connects between the different regions. Most neural signals pass through those network hubs, and therefore, damages to such network hub can be detrimental. On the other hand, such information superhighway was found to improve the performance for artificial neural network, because they allow many layers of neurons to communicate effectively with each other. Because neurons interact by transmitting information, info clustering can apply to find related neurons and the densely connected network hubs. Indeed, the idea of info clustering applies to more general social and information networks where interactions or communications among people generates mutual information. Some examples of online social networks are shown in the figure. They ranges from instant messaging services to online learning communities. Similar to the structure of the brain, it is observed that people segregates into communities, and different communities are also connected by network nubs that reduces the distance between people. There is a small world phenomenon that people are related to one another only within 6 steps. Again, because communication generates mutual information, information theory lies in the heart of the science behind these social and information systems. For instance, info clustering can be used to detect the communities in the social network. Again, because communication generates mutual information, information theory lies in the heart of the science behind these social and information systems. For instance, info clustering can be used to detect the communities in the social network. To cluster objects using information theory, we first associate each object we want to cluster, say I, with all the information that describes it. The information is represented by a random variable, say zi, which can be viewed as a file containing some measurements of the object I. Then, we cluster all the objects based on the mutual information among the random variable ci's. As a motivating example, consider clustering the random variables ci's defined here. The random variables are correlated in the sense that they share some uniformly random independent bits xa, xb, xc and xd. Each random bit is uniformly random between 0 and 1, and the random bits are mutually independent. It is desirable to group z1, z2 and z3 in a cluster because they share a common bit xa. But it is desirable to group z4 and z5 in a different cluster because they share a different independent bit xb. It is also desirable to group z1 and z2 as a smaller cluster because they share the additional bit xd. More generally, let V be a finite set of objects we want to cluster and zv be the corresponding vector of random variables ci for I in V. The set of clusters is defined by the formula of C gamma. More precisely, given a threshold gamma, we consider subsets B of 2 or more elements from V, such that the random variables CB indexed by the elements of B have mutual information IZB strictly larger than the threshold gamma. Out of all such non-singleton subsets satisfying the threshold constraint, we pick the inclusion-wise maximal subsets to be the clusters. For the example we had earlier, if gamma is 0, then we consider subsets with mutual information strictly larger than 0. The candidates are the set consisting of 4 and 5, and any subsets of 2 or more elements from 1, 2, and 3. This is because any such subsets correspond to random variables sharing at least one common bit. We choose the maximal subsets to be the clusters at threshold 0, namely, the set consisting of 1, 2, and 3, and the set consisting of 4 and 5. If gamma is 1 instead, then we want to find subsets of random variables with more than one bit of mutual information. The only choice is the subset consisting of 1 and 2. This gives the cluster at threshold 1. It is clear that, if gamma is larger than or equal to 2, then no subsets satisfy the threshold constraint, and so the set of clusters at threshold value beyond 2 is empty. If gamma is smaller than 0, on the other hand, the threshold constraint becomes trivial. Any subsets share at least 0 bits in common, and the maximal subsets is simply the entire set V. Therefore, the cluster at threshold smaller than 0 is the entire set V. For other values of gamma, the set of clusters can be argued easily to remain unchanged. In general, how do we obtain the set of clusters at different thresholds? The solution depends on how we define the mutual information among multiple random variables. For the earlier example, the correlation structure is simple enough so that we can define the mutual information as the amount of shared bits. However, for general source correlation that may involve more than 2 random variables, how should we define mutual information? Even if we have a meaningful definition of the multivariate mutual information, can we obtain the clusters easily? A brute force search over all possible subsets satisfying the threshold constraints would take exponential time. We have initial success in answering these questions. We discovered a meaningful multivariate mutual information measure, which we call the MMI, that has concrete operational meanings in source coding, network coding, and security problems. The measure has an elegant mathematical structure, called the principal sequence of partitions, that directly gives us the desired clustering solutions in strongly polynomial time as we compute the measure. In the bivariate case involving only 2 random variables, Shannon's mutual information provides a well-accepted measure. H of Z1 is called the Shannon's entropy of Z1. It measures the amount of randomness in Z1. If you think of Z1 as a file, then you can simply think of the entropy as the size of the file after compression. Shannon's mutual information between Z1 and Z2 is denoted by the capital letter I and defined as the sum of the individual entropies of Z1 and Z2 minus the joint entropy of Z1 and Z2. Mathematically, this expression can be rewritten as a statistical distance, called the KL divergence, from the joint distribution of Z1-Z2 to the product of the individual marginal distributions of Z1 and Z2. What it means is that, the mutual information is zero if and only if Z1 and Z2 are statistically independent. To put it simply, if we think of Z1 and Z2 as two independent files that have no shared information, then compressing them together is as efficient as compressing them individually, that is, the joint entropy of Z1-Z2 is the sum of the individual entropies of Z1 and Z2. A well-known extension to the multivariate case is the Watanabe's total correlation. It is defined as the divergence from the joint distribution of the entire random vector to the product of the marginal distributions of the individual random variables. The divergence is equal to zero if and only if the random variables are mutually independent. Although the total correlation is easy to compute and captures the mutual independence among the random variables, we will argue that it fails to capture many other forms of independence relation that arise in the multivariate case. More precisely, consider a partition P of the set V into at least two non-empty disjoint blocks. The product distribution here describe an independent relation, namely, that the agglomerated random variables CCs, obtained by grouping the random variables in C together, are mutually independent. Our choice of the MMI is defined as the divergence to an independence relation, minimized over all possible partitionings of the random variables into two or more groups. It is zero, if and only if an independence relation holds. Note, however, that there is a normalization factor equal to 1 over the number of blocks in the partitions minus 1. This normalization comes into play when the minimum is not zero, that is, when none of the independence relations hold. Explaining this normalization factor turns out to reveal an elegant mathematical structure that, not only gives the desired clustering solution, but also leads to a strongly polynomial time computation of the MMI and the desired clusters. We first start with the well-known Venn diagram interpretation of Shannon's mutual information. Recall that the entropy f a random variable is a measure of the randomness of the variable. If we represent a random variable as a region with area equal to the entropy, then definition of the mutual information can be visualized as the overlap in the randomness of each random variable, which is illustrated by the figure on the right. The Venn diagram can be extended to the eye diagram in the multivariate case. The amount of overlap is McGill's multiple mutual information, which can be obtained by the inclusion-exclusion principle. However, the multiple mutual information can be negative even for a simple example, given by Jung, involving three random variables, namely, when Z3 is the XOR of two independent uniformly random bits Z1 and Z2. Our basic intuition that information is non-negative is violated here. This suggests that we may not treat mutual information as commodity and compute them by adding or subtracting the region of overlap. Indeed, it is well known in network coding that information flow is not the same as commodity flow. It is no coincidence that the example here involve coding, namely, the binary addition of two independent bits. Interestingly, there is a simple fix to the Venn diagram extension, which will also give our MMI expression. Consider rewriting the identity as shown here. We call the left-hand side the total residual randomness after removing the mutual information, because it is the joint entropy minus the mutual information. Each term in the sum on the right-hand side corresponds to the residual randomness of each random variable. In particular, the red region corresponds to the residual randomness of Z1. And the blue region corresponds to the residual randomness of Z2. The identity says that there is no double counting in the residual randomness, that is, the total residual randomness is equal to the sum of the individual residual randomness of each random variable. No double counting here precisely means that there is no mutual information left in the residual randomness, because any mutual information left in the residual randomness will be double counted in the sum on the right-hand side. We call this the residual independence relation, RIR. The residual independence relation can be extended easily to the multivariate case as shown here. P is a partition of V into two or more non-empty parts. The equality says that the total residual randomness with some number gamma removed, is equal to the sum of the residual randomness of the agglomerated random variable CCs, for C in P. It turns out that the MMI is the smallest value of gamma for which the residual independence relation holds for some partitioning of the random variables. The proof is simple. Start with a residual independence relation. Move all the gamma terms to the left-hand side, and all the entropy terms to the right-hand side. The entropy terms on the right-hand side can be rewritten as the divergence. Solving for gamma and minimizing over the set of partitions give the desired divergence expression of the MMI. The normalization factor highlighted in red can now be attributed to the double counting of the mutual information in every agglomerated random variables. This is rather intuitive, since the mutual information is mutual to all random variables, it appears in every agglomerated random variable, and is therefore double counted in the divergence. Indeed, the residual independence relation is more significant than providing a satisfactory explanation of the normalization factor. It leads to strongly polynomial time computation of the MMI and the corresponding minimizing partition. But how do we compute the clusters efficiently using the MMI? Surprisingly, the minimizing partition gives us the desired clustering solution. The minimizing partition achieving the MMI may not be unique, but the set of minimizing partitions form a semi-lattice, called the Dilworth truncation lattice. The lattice structure means that there exists a unique finest partition we call the fundamental partition. Denote the partition as P star of ZV. It turns out that the non-singleton elements of the fundamental partitions are clusters. More precisely, they are maximal subsets of random variables with MMI strictly larger than the MMI of the entire set of random variables. The proof is rather involved, but the idea can be illustrated using the previous example. The MMI is 0 because an independence relation hold, that is, the joint distribution can be factored into a product of marginal distributions according to the partition shown on the right. There are also other independence relations, which correspond to further merging of the agglomerated random variables as shown in the figure. Together with the trivial partition at the top, the set of partitions is referred to as the Dilworth truncation lattice, with the unique finest partition at the bottom called the fundamental partition. The non-singleton elements of the fundamental partition, namely the set consisting of 1, 2, and 3, and the set consisting of 4 and 5, are the clusters at threshold 0, as expected. How about the clusters at other threshold values? It turns out that the clusters at larger thresholds are simply clusters of clusters at smaller thresholds. This hierarchical structure allows us to compute the clusters at different thresholds by computing the fundamental partitions of clusters iteratively. To illustrate this, consider the previous example again. We explained earlier that the cluster at threshold 1 is the set consisting of 1 and 2. However, the set consisting of 1 and 2 is also a cluster of the cluster consisting of 1, 2, and 3. This is because, out of all the subsets of 1, 2, and 3, the only subset with mutual information larger than 1 is again the set consisting of 1 and 2. In other words, the cluster at threshold 1 is simply the cluster of a cluster at the smaller threshold 0. Therefore, starting with the entire set of random variables, we can first compute its fundamental partition to obtain the cluster consisting of 1, 2, and 3. Then, we can further compute the fundamental partition of this cluster to obtain the cluster consisting of 1 and 2. Let us apply the info clustering solution to the problems in bioinformatics and neuroscience mentioned at the beginning. Each random variable zi denotes the expression level of gene I. The expression level of different genes can be measured by the DNA microarray analysis. As shown in the figure, the expressed genes of a sample is fluorescently labeled, and then bind to the complementary genes on the microarray chip. The chip is then scanned by a laser, and the expression level is indicated by the light intensity. Our info clustering turns out to cover an existing gene clustering method as a special case. The method is called clustering by mutual information relevance networks, MIRN. This clustering algorithm first constructs a weighted complete graph on the genes. The weight of edge ij between gene I and j is equal to the mutual information between zi and zj. The mutual information can be computed from the measurements of the gene expression levels. An example of such a graph is illustrated below. The blue edges have weight equal to 0. The red edges have weight equal to 1, except for the red edge 1, 2, which has a weight of 2. Given a threshold gamma, the algorithm filters the edges by removing all edges with weights no larger than gamma. For the example here with gamma smaller than 0, we have the complete graph. For gamma between 0 and 1, all the blue edges with 0 weight get removed. For gamma between 1 and 2, only the edge 1, 2 remains. For gamma larger than 2, all edges are removed. The clusters at threshold gamma are defined as the non-singleton components of the corresponding graphs at threshold gamma. These non-single components are called the mutual information relevance networks. Indeed, the clusters we obtained just now is precisely the clusters we obtained in the previous example by info-clustering. We have shown more generally that info-clustering reduces to the clustering by MIRN when the random variables zi's form a Markov tree. In particular, for our earlier example, it is easy to see that Z1 to Z6 form a Markov chain, which is a special case of the Markov tree. If the random variables do not form a Markov tree, then MIRN solution corresponds to clustering by MMI on the Chaluyeau tree approximation of the random variables, namely, the best Markov tree approximation in terms of the divergence between the true distribution and the Markov tree approximation. However, such an approximation incurs a loss. We can show that the clustering by MIRN fails to capture the higher order statistics beyond pairwise mutual information. As an example, let xi's be uniformly random bits, and define zi's in terms of xi's as shown here. In particular, z3 is the exclusive OR of xa and xb. It can be shown that zi's are pairwise independent, and so all the pairwise mutual information must be 0. The complete graph has only 0 weight edges. Hence, when gamma is larger than or equal to 0, all the edges will be removed, and so no clusters will be returned in the MIRN solution. However, we can show that Z1, Z2 and Z3 indeed share some mutual information, because Z3 can be completely determined by Z1 and Z2. If we compute the MMI of Z1, Z2 and Z3, we get 1/2, which is strictly positive. By capturing this higher order correlation, info-clustering correctly identify the cluster consisting of 1, 2 and 3. Info-clustering can also be applied to study the physical and functional connectivity of the neurons in the brain. With the current advanced neuroimaging techniques, we are now able to see both the physical topology of the brain as well as the functional connectivity of the neurons. The physical interconnections of the neurons can be captured by the diffusion spectral imaging. And the activity of the brain can be captured by the functional magnetic resonance imaging. As mentioned earlier, the brain segregates into densely connected regions. There are also some network hubs that integrates the different regions together. To quantitate the segregation and integration of the brain, some information theoretic measures have been proposed in a framework called the integrated information theory. Essentially, the theory tries to extend Shannon's measure of mutual information to the multivariate case involving multiple variables. Unfortunately, the extension is not rigorous, and some parameters in the measures are chosen in a rather ad hoc manner. Under the info-clustering paradigm, we find a more rigorous way to define such measures. We can think of the connection of the neurons simply as communication channels that generates mutual information. The random variable associated with a neuron or a region of interest can be the set of random variables representing the input and output stimuli. In this way, we can turn a graphical or hypergraphical model of interconnections into a set of random variables for info-clustering. For random variables generated this way, the measure of mutual information has been proved to be equal to the network throughput, or the broadcast rate. The throughput is a max-flow min-cut expression that naturally measures the connectivity of the network, and hence, the integration of the neurons. Info-clustering for this model also reduces to finding subnetworks with throughput larger than that of the overall network. As a simple example, consider an undirected network where each edge is a communication link. The communication rate is specified as the edge weight. The broadcast throughput of the entire network is equal to 1, namely, that 1 bit of message M can be broadcast from node 1 to all the other nodes. This is the MMI of the emulated source, or the integration of the entire network. The red link between node 2 and node 3 is the bottleneck that limits the throughput to 1 bit. To compute the clusters, we identify subnetworks with strictly larger broadcast throughput. For instance, for the subnetwork at the top consisting of node 1 and node 2, the throughput is equal to 3 bits. The subnetworks at the bottom have throughputs all equal to 2 bits. The desired clusters are the maximal such subnetworks. To measure how segregated the clusters are, we look at the ratio of the cluster throughput to the network throughput. A large ratio indicates that the clustering is very clear because a large ratio means that the intra-cluster communication can be much more than the inter-cluster communication, and so the cluster is more segregated from others. In this work, we proposed a new information theoretic approach to clustering biological systems and social networks. In particular, we formulated the info-clustering paradigm and showed how it can be applied to study the human genome and human brain. Compared to the conventional algorithmic approach, info-clustering follows a bottom-up theoretical approach for clustering. Rather than justifying the algorithm purely by data, which was shown to have many issues, we believe it is more important to lay a rigorous mathematical theory before algorithmic simplifications. In particular, the info clustering is formulated in a meaningful way without requiring any prior knowledge of the number of clusters nor an initial solution to start the clustering algorithm. The solution is unique, with meaningful information theoretic interpretations. It also has an elegant mathematical structure for efficient computation. This is the end of the presentation. More details can be found in this paper, recently submitted in celebration of Claude Shannon's centennial. The authors would like to thank Professor Raymond Yern, the co-director of the Institute of Network Coding at the Chinese University of Hong Kong, for his generous support in the research of information theory. The authors would also like to thank Professor Lab Varshney, Rosanna Chan and Chen Change Loy for their suggestions of many relevant works in machine learning and neuroscience, Dr. Javed Ibrahimi, Ravi Raman and Nai Ding for many helpful discussions, Professor Frank Xischang, Prakash Narayan, Devavrat Shah and the colleagues at INC, whose comments have helped improve the presentation. The first author would like to particularly thank Professor Imre Cheeser for the discussion on the divergence upper bound for secret key agreement and the questioning of its tightness. He would also like to thank Professor Li Zhong for his inspiring insights in information theory. Above all, the authors would like to thank Claude Shannon for discovering the theory of information.